Okay, so it's 9:45. Mm, Can you hear me, Nathan?
Yeah, can you hear me?
Yes, we can
Um,
hear both.
Great. So I would like to introduce our next speaker, who is Dr. Nathan Cousins. Dr. Cousins is currently the director of the Molecular Pharmacology Laboratories at the Frederick National Laboratory for Cancer Research. He was the previous co-editor-in-chief of the AGM. We're very excited to have him today. His talk is entitled Strategies for Assay Selection and for the Development of Robust Biochemical Assays. Nathan, welcome. The floor is yours.
Thank you. Thank you for the introduction, Serene. And I'd like to thank Serene for initiating and leading this really fantastic workshop. And I want to thank the organizing committee for inviting me to participate. And I want to thank all of you for joining today. So as Serene mentioned, I'd like to talk about some general strategies for assay selection for high throughput screening assays and really focus on the development of robust biochemical assays. So I'll begin by talking about general considerations when selecting an assay for high throughput screening, and then focus on two types of biochemical assays, those for enzymes and for molecular interactions. So for high throughput screening, you're pretty much using biochemical and cell-based assay formats, and it's a good idea to think about the strengths and limitations of each approaches as you think about how to place these assays in a very complementary way to advance your project. So for example, biochemical assays are less complex than cell-based assays, and the conditions are typically well-defined, which can improve the assessment of structure-activity relationships among the hits. And biochemical assays can be used to determine mechanistic information, such as binding potency or mechanism of inhibition, and they can typically tolerate higher concentrations of vehicle or test compound, which can help you identify hits with weaker potencies. However, due to this reductionist approach, you might be missing some important physiologically relevant features. For example, it's not uncommon to be working with truncated protein. It might lack important post-translational modifications, etc. So on the other hand, cell-based assays are substantially more complex, but the advantage is that your target is presented in a much more physiologically relevant context. But this additional complexity means that there's a greater potential for off-target activities, and it can be more difficult to interpret the structure-activity relationships. Also, if your target's on the inside of the cell, then the compound first has to enter the cell and then accumulate to a high enough concentration in order to elicit a measurable response. So many biochemical inhibitors will not work in cell-based assays. And also you can have test compounds binding to proteins in the serum, which can reduce their effective concentration. So when considering what assays you'd like to use for your high throughput screening project, really depends a lot on your goals. Many times for the primary screen, the goal is to have data that's robust enough to even allow detection of hits from single concentrations of a test compound with strong statistical validation and high enough quality to really allow you to identify structure-activity relationships. On the other hand, the secondary assay can be of lower throughput, it can be more complex, and provide a more physiologically relevant measure of the biology. But you want to think about how you're going to put the assays together so that they can optimally complement each other in order to support the success of your project. So unfortunately, there's no one assay that can suit the needs of an entire discovery project. There's an interdependence among the assay attributes such that the improvement of one is typically at the expense of another. So assay development is kind of a balancing act, and the balance you achieve depends on your goals in developing an assay that's the best possible assay fit for its intended purpose. So I'd like to briefly talk about some different considerations when selecting an assay for high throughput screening. Of course, cost is an important consideration, not only the materials and the reagent costs, but also associated costs. For example, if you're running a radio-labeled assay, the disposal cost might not be trivial. Here's an example from my own notebook of two enzymatic assays that I had developed to consider for primary screen. And as you can see from these assay statistics, both were quite robust and would be suitable for a primary screen. However, the luminescence assay on the left costed 1/3 of the price of the HTRF assay on the right. So a decision was made to use the luminescence assay for the primary screen and then implement the HTRF assay as an orthogonal measure to evaluate the hits from the primary screen. Now this might sound kind of trivial and obvious, but it's really important to think about the types of instrumentation and workflow that you'll be using for the actual high throughput screen. So you might have several assays that you've developed and optimized on your benchtop, but actually performing those assays on the actual instrumentation and using the workflow that you plan to use for the primary screen can provide you with some important insights. Sometimes certain equipment will favor one type of assay approach versus another one, and you'll figure that out. If you can miniaturize your assay, it can reduce the cost of the assay per well and also save some processing time and not having to deal with so many micro well plates. And then you will want to consider the ability to multiplex. Maybe you can't capture all of the biological information you'd like to capture from a single readout. But if you can implement a multiplex readout, it can provide you with more information, which can help you make better decisions on hits versus non-hits. And this little figure shows an example of an enzymatic assay that was developed with a multiplex readout that allowed us to quantify both the substrate concentration and reaction product concentration. And this gave us a lot more confidence in the hits that we identified. And for a cell-based assay, you could use a multiplexing approach, for example, to differentiate pathway modulation versus cytotoxicity. So a lot of things are possible. There's a number of important considerations with respect to reagents. Consistency of the reagents used for high throughput screening of primary screen is very important. So ideally, you would obtain a large quantity of the reagents from a single lot. And this would include enough to run not only the primary screen, but additional follow-up work. And of course, you'll have to make sure that those reagents are stable under the storage conditions for the amount of time you expect the project to proceed. And then validating the reagents is critical as well. So authenticating cell lines, confirming the specificity and selectivity of antibodies, the purity and identity of enzymes and proteins. And fortunately, there's a number of excellent assay guidance manual chapters to provide you with some guidance about those types of validations. And then for high throughput screening, it's not uncommon for reagents to be sat out for a period of time, sometimes at room temperature, for hours. And so it's important to validate that your reagents in your optimized assay are stable for the amount of time and longer that you anticipate needing them, so that you know when it's critical and you need to change out reagents. Here's an example from my own notebook of an enzymatic assay, and I ran this enzymatic assay immediately after preparing the reagents, and then I left them at room temperature for 24 hours and ran the assay 2 hours later and 24 hours later. And you can definitely see some loss in activity, but the data really convinced us that we could move forward with this assay as a primary screen. The potential for assay interference is a really important consideration. For example, fluorescent compounds can interfere with fluorescence readouts. Colored compounds can interfere with luminescence readouts. And so this can increase the number of false positives and false negatives. And fortunately, there is a rapidly growing section of the assay guidance manual to provide you with information about different sources of assay artifacts and interferences, as well as strategies to mitigate them. The format of the assay is another consideration. Primarily for high throughput screening, a homogeneous assay format is preferred, where reagents are added, but there's no wash steps, there's no removal steps. And this is automation friendly, it reduces the variability in hands-on time, and it increases the reproducibility. The It's important to think about the time required for the different assays you're considering, including offline preparation time for the reagents, whether any incubation steps are required, the actual amount of time that the assay will be online, which can vary depending on whether you're going for a single endpoint or maybe a kinetic read, and then the amount of time that might be required for data analysis. For example, for a high-content imaging screen, this could be quite substantial. The signal stability in many ways will depend on the type of biology you want to measure, but it's really important to think about the signal that you want to measure. Is it occurring rapidly and maybe transient, for example, like a calcium flux, or does it occur within minutes or hours? And how long is the signal stable? The longer the signal is stable, the more flexibility it gives you with automated systems, and the more consistency there is among stacks and microwell plates. Different assay technologies and readouts will have different levels of sensitivity associated with them. And so it's important to think about the sensitivity of your assay approaches. For example, luminescence assays are more sensitive than colorimetric assays. Now, a variety of different assay technologies and readouts have been applied to high-throughput screening assays. And this plot shows you kind of a summary of 146 biochemical assays applied to high-throughput screens and reported in PubChem. And in addition to the readouts that you might expect to see, you can also see some that are maybe less commonly implemented, such as nuclear magnetic resonance or electrophoretic mobility shifts. So a lot of things are possible to incorporate into a primary screen. Just keep in mind that all these different readouts have different strengths and weaknesses with respect to their sensitivity, their reproducibility, their susceptibility to artifacts, and of course their cost. Sometimes it's possible to use the same readout for both a biochemical assay and a cell-based assay, and this can provide some continuity as you move between the different formats. Now, your readout of choice might vary depending on whether you want to go with an endpoint read or a kinetic read. So now I'll talk about biochemical assays for enzymes. An enzyme is typically a protein that catalyzes a reversible chemical reaction and is not consumed in the process. In an equilibrium, the rate of conversion of substrate into reaction product is equivalent to the rate of conversion of reaction product into substrate. So there's constant flux in both directions, although the concentrations of reactants and products do not have to be equal. Now, when you're running an enzymatic assay for a high-throughput screen, you don't want to be at equilibrium. At the beginning of the assay, there shouldn't be any reaction product, and as a rule of thumb, you want to avoid consuming more than about 10 to 15% of your substrate by the time you make your measurement. A variety of different strategies have been developed to follow enzymatic assays, primarily focusing on 3 different species in solution. So for example, in green, you can see that it's been possible to develop strategies to follow the loss of substrate as it's converted into product. And for example, maybe having a fluorescent substrate such that you can follow the loss of substrate as this product is being formed. Free enzymes have been followed, for example, maybe by having a labeled substrate and then looking for test compounds that can displace that substrate. Or if there's a known inhibitor that occupies a certain binding pocket and you'd like to identify a different type of inhibitor, you could label the known inhibitor and then look for new test compounds that can displace that, causing a measurable change in signal. And then different strategies have been devised to follow the accumulation of product over time. For example, a labeled substrate that gets converted into a product that's measurable, maybe it's a fluorescent product, or a reaction product that can be acted on by a coupling enzyme in order to produce a signal. Or even a reaction product that competes with a labeled version of itself in order to result in a measurable signal, and this could be like a fluorescence polarization assay. Once you've selected your assay technology and approach, you'll start to optimize your assay, and this involves determining which components need to be included and then optimizing their concentrations, and it can be thought of as an iterative cycle. the assay optimization cycle. And this was actually taken, this figure, from Assay Guidance Manual chapter focused on protein kinases, but it's applicable to many different types of enzymes. And I will be telling you a little more about these different assay components, but I wanted to mention, if you're thinking about getting your enzyme and substrate from commercial sources, I would recommend to try as many different products available as possible before you make your selection. And this is data from my own notebook. I was interested in developing an enzymatic assay, and I compared in one experiment, same assay conditions, 2 different enzymes against 4 different commercially available substrates. And I used an HTRF assay, and I'm showing you here the signal to background values. And what you can see is there's actually quite a range observable from about 1-fold signal to background to 3. And by far, with this assay, the best substrate came from vendor 3, and I ended up choosing enzyme from vendor B. But you can see that it was worth my time to look at all these different options because there was a lot of variation in terms of their performance in the assay. So as you start to develop your assay, you want to begin by gathering as much information as you can from in-house sources, the literature, your own experience, information about the KM, the specific activity, any considerations about the stability of the enzyme and the substrate, and then control compounds that you can use for the assay development and the high throughput screen. And then you'll select the assay technology based on many of the things that I was talking about. And ultimately, you will end up adopting either a direct readout of the reaction product or an indirect readout. Now, if you go with an indirect readout for the primary screen, then you'll want to also develop an assay with a direct readout of the reaction product that you can use as an orthogonal means to validate the hits from your primary screen. So you'll select your initial assay buffer conditions to start assay optimization based on your experience, information from the literature. And if you're working on an enzyme that hasn't been characterized previously and you don't have any information available, you can sometimes, as a starting point, use conditions that have been reported for a homologous enzyme or even an enzyme that catalyzes a similar reaction. Now, of course, buffer is going to play a critical role in the assay performance, and there's a lot of components that would be considered and then optimized in terms of their concentrations. Salt and pH can, of course, both affect the stability and activity of an enzyme, and enzymes will have a pH where they'll optimally perform, and this could vary from physiological pH, but it's important to optimize your assay as close as possible to pH 7.4. And then you might have to include divalent cations, if many enzymes require them. You might consider to add reducing agents. Just keep in mind that although TSEP is more stable than DTT and beta-mercaptoethanol, it's not very stable in phosphate buffer, especially at neutral pH. And if you're working with lysates or purified protein to prevent degradation, you might include chelating agents or protease inhibitors. Detergents are commonly added for high throughput screening assays, buffers, because they can stabilize the assay reagents, and they can also reduce the absorption of enzymes to plastic surfaces of microwell plates or the tubing of liquid handlers, and they can mitigate the aggregation of test compounds. There are a variety of different detergents that people have used. They're used below their critical micelle concentration. And carrier proteins can be used for many of the same reasons. They can also stabilize enzymes that are present at low concentrations. You might need to use a cofactor, and that should be optimized for the best signal to background ratio. Just be aware that the cofactor might actually interfere with your ability to measure the reaction product. So this could also serve as another means to steer you in one direction or another as you're selecting your assay technology. And then there are a variety of different buffers that you might select. Keep in mind that phosphate buffer is incompatible with reactions requiring magnesium and calcium cofactors, and Tris contains a free amine group that can react with enzymes or substrates, and it can chelate metals. So based on all of this, you'll select some assay conditions to start with the optimization, some incubation times, and then the substrate concentration relative to the Km. And your goal is to achieve a balance between the sensitivity of your assay and the signal. Now, even if a Km has been reported for your enzyme and substrate in the literature, you'll want to actually measure this with the exact reagents and assay conditions that you optimized, because the concentration of substrate you select will influence the types of inhibitors you identify. So in this plot, you can see on the y-axis apparent potency, and on the x-axis substrate concentration relative to the Km for 3 different types of inhibitors. So in black is a competitive inhibitor, and the competitive inhibitor will compete with the substrate to bind the free enzyme. And at high substrate concentrations, the substrate outcompetes the competitive inhibitor, and so it will appear to be less potent, and you could miss that in your high throughput screen. Here's an example of a competitive inhibitor in an assay with 2 different substrate concentrations, and you can see an 11-fold shift in potency. Now you could use this to your advantage. Maybe you're screening a kinase enzyme and you want to avoid identifying ATP competitive inhibitors, and so you screen at very high concentrations of ATP. On the other hand, shown in blue is an uncompetitive inhibitor, which preferentially binds to the enzyme substrate complex. And this complex is highly populated at high concentrations of substrate, but at low concentrations of substrate, the primary species present is the free enzyme. And so these inhibitors will appear to be less potent, and you could miss them in your high throughput screen. The non-competitive inhibitor, shown in red, binds equally well to the free enzyme and the enzyme substrate complex. And so the apparent potency doesn't change with the substrate concentration. So if you wanted to be optimally sensitive to all the different types of inhibitors, you would choose to screen at the Km concentration of the substrate. So how do you measure the Km for your enzyme? You begin by setting the substrate concentration high above the Km value and performing a titration with your enzyme, and select a concentration of enzyme that's within the linear range. And then you perform this experiment that's nicely laid out in an assay guidance manual chapter called The Basics of Enzymatic Assays for High Throughput Screen. You begin with that optimized enzyme concentration that I just described, and then you'll measure the initial velocities with a range of substrate concentrations spanning 5-fold the Km to 1/5 the Km, with an equal number of substrate concentrations both above and below the Km. Now, if you don't know the Km, this might have to be an iterative process until you feel confident that you're able to adequately measure the Km. But you take these initial velocities, these slopes, and you plot them against the substrate concentration, and you fit that data to the Michaelis-Menten model in order to measure the Km value. Now, if your data do not fit to the Michaelis-Menten model, it could be due to issues with substrate solubility. And in that case, you might go back and optimize the conditions a little more to hopefully improve the solubility of the substrate, or even if it's possible, maybe replace that substrate with a different one. Now, what if you have 2 substrates? In that case, you would begin by setting the second substrate concentration high above its Km value and repeating that same series of steps I just described in order to measure the Km value of the first substrate. And then with that in hand, you'll set that concentration high above that value of substrate 1, and then you'll vary the concentrations of substrate 2 the same way you did in order to measure the Km value for substrate 2. And then once you have the Km value, you'll go back and select a substrate concentration based on what I had described earlier. So depending on your goals, do you want to be optimally sensitive to the different types of inhibitors? Do you want to bias yourself against identifying certain types of inhibitors? So select the concentration of substrate that you think is ideal, and then repeat that enzyme titration curve and confirm that it's still in the linear range. And then you're ready to start testing any available control compounds that you might have. So let's look at some different types of data. This again is from the Basics of Enzymatic Assays for High Throughput Screening chapter. And this plot up here shows an example of saturating the detection system at only 20% capacity of reaction product. And this is just to point out that even if you have a well-optimized assay, it's critical to look at the detection system and kind of confirm that you're able to measure the reaction product across the whole range that you plan to measure it. Down here on the lower left is an example of a well-behaved enzyme. You can see with the 1x concentration, initially there's a linear phase of the reaction, and as substrate is depleted, it plateaus. And if you double or half the concentration of the enzyme, you see corresponding increases or decreases in the reaction rate, but it always plateaus at the same place. And you can contrast that with the example on the right, where the height of the plateau depends on how much enzyme you add. And so this would indicate a loss of enzymatic activity, and it could be due to the enzyme absorbing to the surface of the microwell plate. Maybe the solution conditions make it unstable. So you would go back and try to further optimize the solution conditions to hopefully make that enzyme a little more stable, and your data will look a little more like this scenario on the left. Now, it's important to make your measurement for high throughput screening during the linear phase of the reaction. And that's, I think, really well illustrated by this figure here from Robert Copeland's Evaluation of Enzyme Inhibitors in Drug Discovery book. So this plot over here shows an enzymatic reaction. We're looking at the product generation over time. And this data with the filled circles shows the reaction when it was treated with vehicle compared to these data with the open circles that shows the same reaction in the presence of an inhibitor that reduces the activity by 50%. And if you look at this data inset here, you can clearly see that during the linear phase of the reaction, there's a 50% reduction in the reaction rate. And it's also seen over here where apparent percent inhibition is plotted versus time. But what happens is, as it leaves this linear phase of the reaction and it starts to plateau, there's the apparent percent inhibition drops. And many high throughput screening centers will set a threshold for hit identification at maybe 30% inhibition. And so what that tells us is that if you made your measurement after an hour, you would no longer even identify this inhibitor, even though you can see during the linear portion of the reaction, it's clearly inhibiting the enzyme. Another thing to keep in mind is that the most potent compounds that you can possibly measure with your assay will appear no more potent than half the concentration of your enzyme. So, for example, in this assay here, the enzyme is present at 50 nanomolar. So even if you had a picomolar inhibitor, the IC50 would appear as 25 nanomolar. And so it might be the case that you will optimize an assay for a high throughput screen to identify hits. And then later when you're supporting lead optimization, where you're really looking to distinguish more and more potent compounds, then you would re-optimize your assay later, possibly with more sensitive readout and a much lower concentration of enzyme. The goal is to establish the most robust assay possible using the most appropriate controls that you can find. And so what makes a control appropriate is it should really resemble the type of inhibitors or molecules that you're hoping to find within your chemical library. And then it's really important to think about the effect of your vehicle, which is often DMSO, on assay performance. Sometimes it can actually have a substantial impact. And here's an example from my own notebook. You can see this is a cell-based assay with an alpha LISA readout. And it was done in 1536 well format. And I'm showing you the data as a heat map. So purple is low signal and red is high signal. And the purpose of this experiment was to determine a Z prime factor. And so the first 4 columns were treated with an inhibitor that reduced the signal by 100%. And then the remaining wells were treated with vehicle. And the expectation would be that this would be a high signal and it would be uniform throughout the plate. But what you can see is that there was a drop off in the signal on the right side of the plate. Because this was a cell-based assay and it sat in the incubator, one of the first thoughts we had was maybe this was an issue due to a temperature gradient. We looked into a lot of different things, but one of the things we tested was liquid handling. And so the vehicle and test compounds were delivered with a pin tool, and so we used that same pin tool to transfer dye and to buffer. And you can see, this is the data down here, you can see that there was actually more dye transferred to the right side of the plate here. And this pattern kind of resembles this pattern above the initial finding. And it would suggest that more DMSO was actually transferred to these wells over here and that lowered the signal. And sure enough, if you do a DMSO titration, you can see that just going from 0.3 to 0.6% DMSO nearly abolished the reaction signal. So now I'll switch gears and talk a little bit about molecular interactions and assays for molecular interactions. One common approach is enzyme-linked immunosorption assay or dissociation-enhanced lanthanide fluorescence immunoassay. And these are sandwich-type approaches that can be applied with purified components or complex samples such as lysates. They have typically very high sensitivity, excellent dynamic range. Earlier I was mentioning that homogeneous assay formats are preferred for high throughput screening. This assay format does require wash steps, but one advantage of that is that you end up removing the test compounds, and so you ultimately have lower interference with this assay format from test compounds. A commonly used approach for proximity assays, looking for the proximity between 2 entities, is time-resolved Forster resonance energy transfer, or TR-FRET, or HTRF. And this proximity technology involves a lanthanide donor with a long-lived emission. And when it becomes proximal within about 20 nanometers from an acceptor, there's a long-lived TR-FRET signal that can be measured. And the advantage here is that when you initially excite the donor, you can also end up with unwanted sources of fluorescence coming from the matrix, from test compounds, but that drops off faster than when you make your TR-FRET measurement. So this ends up being a very clean measurement. The assay itself can be very sensitive with an excellent dynamic range. It's a homogeneous format. It's amenable to multiple time point reads. And in addition to being able to measure the TR-FRET signal, you can also measure the donor emission. And this allows you to identify some types of test compounds that can interfere with your assay. Another commonly applied approach for molecular interactions is fluorescence polarization assays. And they can use a fluorophore as a ligand or a fluorophore conjugated to a ligand. And when this fluorophore is excited with polarized light and it's rotating during its excited state lifetime, when the emission occurs, it's depolarized. Unless the fluorophore or fluorophore-bound ligand binds a larger molecule, and it slows down its rotation, and then the emission becomes more polarized. And this can be used as a readout of binding. And it can be applied to purified proteins, peptides, small molecules, It can be a very sensitive assay, excellent dynamic range. It's a homogeneous format, amenable to multiple time point measurements. There are different considerations depending on whether you're interested in a very strong affinity interaction or a weaker affinity interaction. With a weaker affinity interaction, it can require so much material that it can become cost prohibitive. On the other hand, with strong affinity interactions, you have to use really low concentrations of probe, so you can struggle to get enough signal, or fluorescent test compounds, which are present at much higher concentrations, can swamp out your signal. Fortunately, there are a range of fluorophores that have been applied to fluorescence polarization assays, and they include different fluorescence lifetimes, which allows you to connect them to different size ligands. And a survey of 213 annotated fluorescence polarization assays from PubChem that have been used in high throughput screens shows quite a range of different fluorophore types and colors, including green, yellow, orange, and red. I would say a word of caution from NCAT's experience is that it's worth your while to evaluate multiple fluorophores before you settle on one for your assay. And this striking example here, really goes to show that this was a protein peptide interaction assay. And you can see that when we used Bodipi Texas Red, we couldn't even measure binding. On the other hand, Tamra gave us the best dynamic range. So it was definitely worth trying multiple fluorophores there. And then sometimes after you've optimized your fluorescence polarization assay, you can even improve the data more, expand your dynamic range by optimizing the excitation and emission wavelengths. And you can see that here if you compare these red squares to the blue circles data. Now I'd like to combine some of the things that I just mentioned and introduce a few more concepts in the context of a case study based on a collaboration I had a couple of years ago with someone who was interested in developing a biochemical assay to identify small molecule inhibitors of a peptide protein complex from a high throughput screen. And this peptide protein complex only occurred in the presence of an additional ligand. And when we first started talking about the project, my collaborator showed me this data that I'm showing you in the center of this slide. And you can see in the consistent concentration of Tamra-labeled peptide, as my collaborator added additional protein, you can see an increase in the polarization signal, indicating binding in the presence of this ligand. And so they wanted to convert this into an assay for high throughput screening to identify small molecule inhibitor. At NCATS, we successfully miniaturized this same assay into a 1536 well format. And you can see that based on the assay statistics, this would be suitable for high throughput screening with a Z prime factor of 0.5. However, it required almost 2 milligrams of protein per plate, which was incredibly laborious for the lab and somewhat, it was fairly costly. So they wanted a more sensitive approach. And we turned to a technology called AlphaLISA from PerkinElmer. And this is a bead-based proximity assay involving a donor bead that's excited with 680 nanometer light. And this causes the generation of singlet oxygen that can diffuse to a distance of about 200 nanometers in solution, exciting an acceptor bead, resulting in a chemiluminescent emission of about 620 nanometers. Now this is among the proximity technologies, this is probably amenable to the widest range of molecular sizes. It's typically a very sensitive and robust assay approach. It is light sensitive, that's one caveat. But these beads can be decorated with many different molecules on the surface. And we use that to our advantage to take this highly specialized fluorescence polarization assay that we had developed and just using out-of-the-box reagents converted to this AlphaLISA technology. So I had mentioned that my collaborator was using a Tamra-labeled peptide. We simply ordered the peptide biotinylated, and it was able to tightly interact with streptavidin conjugated donor beads. And then my collaborator had already been purifying the protein component using a polyhistidine tag, and nickel chelate acceptor beads are available, which bound the protein component. So here, we were able to take this very specialized set of reagents for a particular assay biology and just convert the technology with out-of-the-box reagents. And we wanted to know what concentrations of biotinylated peptide and protein to use, so we performed a titration in a 1536 well plate with 16 replicates per condition. I'm showing you the raw data, and it's colored as a heat map with red for a low signal and green as a high signal. And I want to draw your attention to this column here with 9 micromolar biotinylated peptide. And you can see that as we increase the concentration of polyhistidine tag protein, there's an increase in signal, and then that's followed by a decrease in signal as we keep increasing the concentration of protein. And this is actually due to the hook effect. And this is not just common to alpha-LISA assays, but it's common to any immunosandwich type approach. And what happens is at low analyte concentrations, all of the analytes will result in proximity to the beads, giving a signal. And as you increase that analyte concentration, you get a corresponding increase in signal, up until the point where you saturate your detection system. And beyond that, as you continue to add analyte, it actually outcompetes these signaling competent complexes, and that's what drives your signal down. So earlier when I was talking about saturating the detection system, this is another way to do that, so watch out for this one. So going back to that data I showed you, we calculated signal to background values and Z prime values, and we're ready to move forward with assay optimization, and we needed a control inhibitor. And we didn't have a small molecule. That was the goal of the project. So we ordered a non-biotinylated peptide to use as a competitive inhibitor of the biotinylated peptide. And the anticipation was as we increase the concentration of the non-biotinylated peptide, we should get a dose-dependent decrease in signal. But what happened was we actually saw the opposite. You can see that the signal really blew up with high concentrations of non-biotinylated peptides. So we looked at the solution conditions, and it turns out that actually the buffer pH was aligned with the isoelectric point of the protein, which is the pH at which the protein is least stable. So we hypothesized that this really large signal was due to aggregation of the assay components. We went back and adjusted the assay buffer 1 pH unit more basic, repeated the experiment, and then saw a nice dose-dependent decrease in signal, as expected, with higher concentrations of non-biotinylated peptide. The IC50 value matched the affinity that we were expecting with the molecular complex. And nicely, this optimized assay only required 61 micrograms of protein per 1536 well plate, whereas I told you that the fluorescence polarization assay required nearly 2 milligrams. So we used this assay for the high throughput screen, but then we were able to use the fluorescence polarization assay as an orthogonal approach to validate the hit compounds. And then just one final note is, in addition to optimizing your buffer and the fluorophores and the different components of your assays, sometimes it's possible to improve your assay just by optimizing the equipment settings. And here's an example. This is, again, a 1536 well plate shown as a heat map. Red is high signal, purple is low signal. You see this gradient here. And when I flipped the plate, the gradient stayed in the same position, indicating it was with the plate reader, not the plate. When we optimized the settings of this instrument, you can see the percent coefficient of variation improved by 75%, improving our Z prime factor from 0.7 to 0.9. And so that resulted in a lot of improvement in the assay performance. And finally, I took most of these examples from the NCATS Assay Guidance Manual. It's an excellent resource for assay development best practices. It's freely available worldwide through this link here. You can email the editors if you have questions, if you'd like to see additional content, if you'd like to contribute content, and you can follow the Assay Guidance Manual on Facebook and LinkedIn. And I want to thank you for your attention, and I'd be delighted to take any questions that you might have.
Thank you very much, Nathan, for that wonderful presentation. So let's look if we have questions. Yes, I do see a question. So how can you reduce, this is from Core Carter, how can you reduce assay interferences when the purity of synthetic or isolated natural products are not quantified by NMR? And then he follows it with, how many times would you recommend to test your assays to be considered significant?
Yeah, so with the first question, we're going to hear a lot more about assay artifacts and interferences in the next 2 days, I suspect. There's a lot of great information in the assay guidance manual about this, but I think the question about the natural product is really important, because there can be a lot of additional molecules in a sample in addition to what you think. And this is true for synthetics too. There can be a lot of contaminants, and they can cause interferences and artifacts. To be very honest, it can be challenging, even just for synthetics. And so a lot of work has to be done in order to confirm that the particular structure that you think is modulating the biology is actually modulating it. I think it is much more challenging for natural products, and I'd be interested if any of the other panelists wanted to add something to that. And then the other question about how many times do you have to test your assay in order to confirm that it's validated? I think that it depends in part on the assay statistics. They'll really tell you the performance the way you're looking at the high controls and the low controls. And of course, you want to use the most physiologically, pharmacologically relevant controls that you possibly can. That's critical, because if you're using kind of an unnatural control that's not similar to the types of compounds you're really hoping to identify from your chemical library, it can really sometimes mislead you in terms of the type of activity that you're looking for.
And we will be having a whole session on statistics tomorrow morning, so hopefully that can help as well understand to know what assays data is significant or not. Let's see, any other, I don't see any other questions in the question and answer, and I'm going to check the chat box now. I do not see any further questions, so please type in your questions. Okay, so here's another one, Nathan. Would you recommend a TR FRET or alpha screen assay in cellular screen? This is from Gizem.
They're both excellent approaches. I'd actually recommend both. It's ideal whenever possible to use multiple technologies because they won't typically be sensitive to the same types of artifacts and interferences, and they'll have different levels of sensitivity, and sometimes you can learn things. So the more assays that you can incorporate and develop, the more confidence you can have in what the data is telling you.
Thank you, Nathan. Checking if there's more questions coming in. I do not see any further questions in the chat box, nor the question Q&A. Any more questions, please type in your questions now. If the panelists have any questions, or would you like to add on any of the discussions, please feel free to unmute yourself and discuss with us.
Serene, we're waiting
Yes.
on that. Hi, this is Abigail. I just typed into the chat box that there's a few spots available for the lunchtime roundtables. So anyone who is still interested should chat me a message which roundtable they're interested in. We'll try and get them in. And then after the final questions from Nathan, we'll have a break until 10:45, and then we'll have Terry.
Okay. All right. Thank you, Abigail. So I do not see any further questions for Nathan. So thank you very much, Nathan, for a wonderful
everyone.
presentation again.
Thank you,
Thanks.
Serene. Thanks, everyone. Really looking forward to this event. A really exciting agenda ahead of us. Thanks, everyone.
Awesome. Thank you, Nathan. So we do have a break, as Abigail said now, and we have about 10 minutes break, and we will reconvene at 10:45 to hear from Dr. Terry Reese. So if you have any questions during this time, please keep typing your questions in the chat box. And as Abigail said, you can type in whether you would like to be added to the.
Yes, that's right. People can send me a chat message and we'll try and fit you in.
All right, wonderful. Thank you very much.
Thank you, everyone.